I wanted to just show everybody the sign-up sheet that I mentioned that set up yesterday. I had thought I had done this in your Blackboard section, and this morning I realized I had set it up in one of my other classes, and so it wasn't visible to you. And for whatever reason, I didn't get around to copying it into the different courses. So it's there now. And you can click on this and it'll take you to this page here. And then you want to type your name in the column for this section, B20. Type in your name for the connective tissue that you want to do. I'm giving everybody the power to edit this document, which is kind of scary because obviously you have to type something in there, but also it means you could potentially erase somebody else's name and type yours in. Do not do that. If somebody else has signed up for something and which you would hope to do, it's first come, first served. So do not take somebody else's away. And to alleviate some of the fear in this kind of shared document environment, it saves history. So if you did go in there and type over somebody else's name, I'll know that, okay? And you will suffer my wrath if you do that. So please, if there's a spot already filled for what you wanted, don't sign up for it on top of that person. If you really, really wanted to do something, and there's nothing else for you to do, ask me, and there might be an alternate version of that tissue that I can have you get copies of. But I don't expect that to happen. I think there's plenty of spots for everybody. And also I want to point out, pay attention to what you were assigned in the first section because you might want to sign up for something that is using the same slide. So for instance, slide R11 is for the keratinized stratified squamous epithelium, because that's in the skin. R11 is used again for dense irregular connective tissue, which is another part of the skin. And I thought it was used somewhere else. Oh, that's right. No, never mind. So keep that in mind. It will potentially help you do this stuff a little bit quicker if you want to. But again, don't sign up on top of somebody. If you go to sign up for something that uses your slide from the first set and somebody else has picked that, let me know and I'll see if I can convince that person to switch so that you can use the same slide. But don't take matters into your own hand because I definitely will not allow you to do that. So keep that in mind. Also, I think I mentioned right at the end, people weren't aware of this, the letter in each of these slide designations refers to the box that it's in. So R for red, G for green, and Y for yellow. None of the slides we use in this come from the blue box. There's a question mark next to this one for dense elastic connective tissue. If you sign up for that, I'll try to remember to verify that slide number when I'm in lab this afternoon. But if I don't, it's actually for the aorta. And so if you're doing that, it might be slide 21 in the yellow box or something like that. I don't remember. So when you go to do that, be sure that you get the slide that's the aorta, even if it's not slide 20 in the yellow box. And hopefully, if you don't remember that much of it, at least the question mark will make you ask me a question about that. and I'll verify that you're using the right slide there. But it's 20 or 21, maybe it's 19. I know it's right around there, but it's specifically it's the aorta that the slides up. If you choose to do that one. So for next Monday in lab, you'll want to have made a choice for which tissue you want to do before you come in. And it'll be an unstructured lab time. You'll just come in and get your scope out, find your slides, look at the scope, ask me to look at what you found because I want to be sure that you're looking at the right thing. If you want to, before you come in on Monday, do a little research and make sure you recognize what the slide is. I mean, what the tissue looks like. That's fine. And consult with me. I want to okay what you're looking at before you do it, so you're not taking the wrong picture. And then you'll take your pictures on the digital scope. If I'm available, if I'm not helping somebody else with their scope, I'll be there to answer questions about how to use the scope. If you're not comfortable with it, wait till I'm available. And when you're done taking your pictures, you're welcome to leave on Monday. You don't have to stay there for the whole time. If you get in right away and take your pictures and you're done by 10:30, you can head out. Okay. I do want some quality to this. So don't run in, take some quick crappy pictures and walk out. Make sure that I've okayed that you're taking pictures of the correct thing, and that's why you want to get your scope out to look at it. And then when you're taking the pictures, remember you're taking pictures for you and everybody else to work with, to study from. So they need to be in good focus and the lighting quality has to be pretty good. A few people stuck around to take their pictures at the end of class on Monday, that'd be yesterday. I haven't put them in that folder, but I will, maybe I'll get it done before I start my lab at 1 o'clock today. So you can look in there and see what other people did if you want to get an idea of what the lighting and focus conditions should look like. But hopefully there will be other people around and you can consult people and say, does that look in focus to you? That kind of thing. So that's what we're going to be doing next Monday. And you'll want to, before you come in, choose the tissue that you're going to be doing. So to do that, remember in the tissue level organization folder, there's the tissue slide sign-up sheet. So select that and put your name in. I don't think for you guys it's so obvious because your names are already in that column for the first part, but I do want to be sure that everybody's paying attention to which section they're signing up for. So I've listed them here in the instructions. Any questions about what I'm asking you to do for that? Okay. Now, this week we're going to be looking at the tissue in lecture. I have put up, oh, that's right, I haven't finished putting this up. But I do have the epithelium and connective tissue materials up. Which I wanted up in time for you to look at them. Well, the epithelial one for you to look at before you came in to the lab yesterday. Connective tissues I got up right before lab, but you'll be working on connective tissues next week, so you'll have plenty of time to look at that. I have to do the other tissue presentation stuff, and I'll add those in here. But I do want to spend a little bit of time today talking about some of these things. So we'll go there. Now, for Thursday, there's a chance that I have a conflict. And I hesitate to say this. I was really trying to get in touch with the person who this is being scheduled with to verify when I was going to be done and if I'd be back here by 11. So what I have to do is to tell you, I don't know if we're going to have class on Thursday yet. I hope we are, but I'll send out text messages to people when I know more definitely. Hopefully I'll know tomorrow, so I'll let you know. But if, I don't know, this is the only class you come in for on Thursday or something like that, if you want to, before you leave home, check with me, and hopefully I'll know by then. I've told everybody, but if Thursday morning you say, do I have to come in for class today, text me and I'll make a decision then. And what I might end up doing is canceling class. If I cancel class, what I'm supposed to do is to give you an equivalent amount of work to do to take up time, like take home homework or whatever. I don't do that because there's plenty of stuff that you do on your own time with this class, the way it's set up. So if I do happen to need to cancel class on Thursday, if anybody asks, I gave you all this work to do, okay? But really I gave you work to do already, so I don't have to assign special stuff. That'll be fine. But if I can make it in for class, I would like to still potentially have class. And I'll let everybody know about my decision of that when I hear from this person that I have to schedule this thing with. I'm supposed to do something at 10 o'clock on Thursday, and she hasn't gotten back to me to tell me when it's supposed to be over. So I'm hoping it'll be over by 11, but if not, I might cancel class for you guys, which I'm sure will break your hearts. So before we get underway with stuff today, I just want to check, hopefully you have all, if not some of you, looked at those presentations for the tissue stuff. Are there any questions people have, things that you want clarified of how I went over it? Everything crystal clear? All right. I'm going to probably ask that kind of question repeatedly. I am just sort of rolling out this approach. As I'm doing it, I'm basically sitting at my desk and I have this fancy scope microphone in front of me. And I just have the presentation up there and I go through it and I record a voiceover for each piece, which is essentially kind of what I do in lecture. Except that I get a little bit overly wordy in lecture, so I try to trim things down a little bit. Yeah.
Do you have notes you read from, or is this just all right out of your head?
Off the top of my head.
That's pretty cool.
I've been doing it for like a decade or more, so it's not that hard. What's that?
About regeneration, cell regeneration, how that happened.
Regeneration.
Like when the cell die and then regenerate.
Is there a particular cell type that you're asking about?
No, it's any cell.
Okay, because it's different for different types of cells. And sort of in that context, let me go to something that I don't have a presentation on yet. So I can kind of have it in the context of this chapter's material. Okay, so just a second as it's loading the chapter. But there's a section, and why the heck is it so big? Obviously the book has been updated, and so the link there is a little old. So let me just quickly fix that link. Wrong place. So in the tissues section, there's a section on tissue injury and aging. Oh, that's right. I was just doing an ANP2 presentation, and the picture about white blood cells looks very similar to this picture, except that it's oriented 90 degrees. I can't remember, counterclockwise or clockwise. And so I was thrown for a second, kind of like, this isn't the right picture. But no, this is not the um, blood vessels picture. So this is about how tissues heal, and it's in the context of a cut to the skin. And in that, I think I'll address part of what you're asking. And the internet's really slow here, but oh, you can do it. So first off, I've said this a couple of times, well, in the presentation, maybe not out loud. Epithelial tissues are avascular. They don't have a blood supply themselves. The cells are very, very close together. I like to use a brick wall, mostly because we have brick walls all over the place in the building. But if you imagine each of those bricks is a cell, there's no space for anything to get in between the bricks except for the mortar that's holding them together. And so the blood vessels are usually found adjacent to the basal surface of the epithelium. Like this conduit here has the wires that go to the bone box or whatever, or this is the electrical side. Imagine those are blood vessels that are on the basal surface of the epithelium, and the nutrients that are in the blood will diffuse through the tissue, and wastes from the tissue will diffuse out to the blood. And I mention that because, well, one, I'm trying to kind of kill some time while this figure's loading, and two, because I think it's important to point out. In this, there we go, in this injury, the cut is going to go through the epidermis, which is the outer layer of the skin, and down into the dermis, which is the deeper layer. The epidermis is epithelial tissue. It's specifically stratified squamous epithelium. And the dermis is connective tissue. Mostly it's dense irregular connective tissue. Okay. There's a few other things going on there, but it's mostly dense irregular. And in that dermis region, the connective tissue, there's a lot of blood vessels. And those blood vessels nourish that tissue itself, but also the epidermis, the epithelium on the outside side. And so if you cut through the skin down to the dermis, you're going to damage the blood vessels down there and they'll bleed. Okay, so that's what we see here is the accumulation of the spilled blood, which is going to clot and form the scab that we see there. Okay. And epithelial tissue and connective tissue respond to damage differently. Okay, so we see that the epidermis and epithelial tissue is completely severed. Okay, this side and this side are separated from each other. What's going to end up happening is the individual cells, which make up the basal layer of that epithelium, are going to start dividing more rapidly and replace the damaged tissue. So I think that's kind of what you mean by regeneration. For an epithelium, there are going to be cells that are specifically tasked to divide and make new epithelium. For simple epithelia, it's really just any cell in that layer. For stratified epithelium, it's, well, it varies from example to example, but this is the most common one in your skin, and it's the basal layer that divide and they sort of fill in where there's a gap. So in the second picture, we see the epidermis is now continuous here, right at its basal level, and it's going to eventually sort of fill in that space. And so those cells in an epithelium just automatically they're always dividing and replacing old cells, and so it will just grow in and fix it up. With the damage, it sort of stimulates faster regeneration of those cells to grow back in. With the connective tissue, it's a little bit different. It's almost like connective tissue takes everything very personally. Okay? So when there's damage to connective tissue, a type of cell called a fibroblast will move in to repair the tissue. And fibroblasts are called fibroblasts because they make fibrous proteins like collagen. And so it's sort of like they're offended that the tissue is damaged. So when they grow back, they make it stronger and they lay down more collagen fibers, which is what we see there. It's sometimes referred to as granulation tissue because of what it looks like under the microscope. More often, you'll hear it referred to as fibrous or scar tissue. And it's just that there's more collagen there helping to hold the tissue together. It's like, sure, you cut me once, but you're never going to get to do it again. It just makes it stronger. And we see that in all sorts of connective tissues. This is the dermis of the skin. You break bones, you have bone scars, and just because the collagen grows back stronger. Okay. When you see a scar in the skin, you're actually seeing the collagen that's deep underneath more than anything affecting the epidermis. Okay. The epidermis is only like a millimeter thick, maybe 2 in certain places. So it's very thin, and you can see evidence of what's going on with the connective tissue below it. So the scar tissue is really in the dermis, and it's because of all of this fibrosed scar tissue growing in to make it a little bit stronger. Given enough time, that tissue is likely to sort of get back to normal, but we're talking about decades and decades of time. So a significant scar will always be there. But if you just make a little cut in your skin, so if you think about the scale of everything that's drawn here, this is actually a fairly small cut. And given enough time, a small fibrous area like that will eventually get back to its normal configuration. The excess collagen will be rearranged, and some of it will be broken down. It'll be like it was before. But a big scar is going to stay for a very long time because it's so big, it's just never going to quite get reorganized back to its original shape. Okay. The fibroblasts will divide and regenerate the tissue and fill back in. It's just that the tissue sort of is trying to do its job a little bit better. Like I said, I always kind of characterize it that it's sort of taking it personally that, you know, it was cut and it's never going to let that kind of failure happen again. Sometimes that actually is kind of a problem because the difference in the density of the tissue where the scar is versus the regular tissue actually creates a little the word that's coming to mind is sensitive, but that's not the word I mean. It's not that you can feel a difference, but rather it's vulnerable. That's the word I want. So that the change in density between tissue there actually makes that little part right there a little more likely to get damaged than the regular tissue over here or the scar tissue. In bone scars, that's most evident. When people break their bones in the same place, they're actually not doing that because where the first break was, there's a scar. It's stronger right there. But where that bone scar meets up with regular bone, there's a little bit of vulnerability right there. And so when you break a bone in the same place, you're actually breaking it right next to where it broke before. And there's just a slightly better chance that happens because of the presence of the scar tissue there. Now, if you want your bones to never break, if you completely break them over and over again and they just heal back to just be one big bone scar, then you're protected against that. But I suggest you don't try that because that can be quite unpleasant to live through. So now those are two types of tissue. There's also muscle tissue and nervous tissue. And nervous tissue is special. So I want to go into that a little bit in depth in a second. Muscle tissue is often regenerating. And to some degree, it's kind of like connective tissue, maybe in between connective and epithelial tissues. If you damage muscle tissue a little bit, it'll grow back and you would never know that it happened. It does, in fact, grow back a little bit better than it's supposed to, or better than it was before. And that's the basis of going to the gym and working out. You're trying to push your muscles to the point where you damage them a little bit. And when we look at muscles in a later chapter, we'll talk about the structure a little bit. But at the molecular level, you're actually damaging the muscle tissue. But it grows back and repairs that damage and actually will increase its ability to generate force above what you asked it to do. So when you go to the gym, you're trying to work out until you go a little bit beyond what your muscle is capable of, and you start to damage the muscle, and the muscle will repair itself and be able to do whatever you ask it to you before. And then you push a little bit further, you damage a little bit more, and it grows back and it can do what you meant to do before. That's mostly happening within the cell structure, you're not damaging whole cells, but you're damaging the proteins within it. There are stem cells in muscle tissue that can regenerate damaged tissue to some degree, but when you go too far, then there's connective tissue in muscle also, which will form scars and really kind of mess everything up. So if you've ever seen somebody with significant damage to muscle tissue, there are muscle scars, and that's because of the connective tissue in muscles, not the muscle tissue itself. But it can interfere with the ability for the muscle tissue to regenerate back. Yeah.
If somebody breaks a bone and they don't do what they're supposed to do to like get it fixed, as far as like breaking it back in place or whatever, and it just fuses the way it is that worse than?
Sure. I mean, if you change the anatomy of your body so that it's not the way it's supposed to be, optimized to its function, then it's going to cause problems. I actually have some experience with that. I broke my leg when I was a kid, and there are two bones in the lower part of the leg. And when they broke, they healed back like this. And so there's just this big knot of bone tissue. And there's a 10-degree angle in the bone. And at the time when I broke my leg, there was a bunch of other things going on. And so the orthopedics department didn't really do a great job at making sure that it healed back properly. I don't quite know who to blame. I'm just not the sort of person that's going to, go out and start suing people for that. But it's very suspect. I don't quite know why it went that way. Later on in my life, I went and had that repaired, where they basically surgically broke the bone again, and they put a rod into it, so it healed back the way it was supposed to. But in between, when I originally broke the leg and when I finally got it fixed, that 10-degree bend actually put a lot of stress on my knee that's not supposed to be there. So I have some problems with the cartilage in my knee. So yeah, if something like that happens, it's definitely going to affect what's going on in your anatomy and create problems. Now, nervous tissue is special. We separate nervous tissue anatomically into two regions, the central nervous system and the peripheral nervous system. The central nervous system is the brain and the spinal cord, and the peripheral nervous system is everything else. Regeneration is different between those two different areas. Now, when we talk about damaging nervous tissue, we're usually thinking about cutting nerve fibers. Okay? And in the central nervous system, nerve fibers do not grow back. They're actively inhibited from regrowing. And in the periphery, they do grow back, and they're actively supported in regrowth. There's been some recent work showing how we can do some things in a spinal cord injury to help things grow back. I think the original announcement came out, what is this, back in the spring, but I've seen a few news sources pick up the story recently. So there's this hint that we might be able to help regenerate spinal cord damage or something like that by introducing some of the signals that are present in the periphery into the central nervous system to get things to grow back. The problem with it, and the reason why the central nervous system doesn't grow back, is that the connections in your nervous tissue is very important. You've spent your lifetime building those connections in how you learn. And if you just let things regrow back after damage, there's a high probability those connections will get mixed up. And it would cause forgetting, and you might not be able to control the muscles the way that you're supposed to, the way that you've learned to. So they don't grow back in the central nervous system on purpose. And with medical advances, hopefully we can find a way to make sure that they grow back appropriately so that we don't mess things up like that. Now, as far as just cellular regeneration, which kind of goes back to your original question, it had always been the expectation in the brain, especially, that you were born with all the neurons, nerve cells, that you're going to need, and they last for your whole life. But I can't remember exactly how long ago it was. It was when I was in school. So sometime in the last 3 decades, New data surfaced to show that there are new nerve cells that grow in certain parts of the brain. And there's apparently a fairly active growth in certain areas, and in other areas that just don't have any new growth. So it's, again, kind of special where it happens and where it doesn't. But that regrowth doesn't seem to be to handle injury, but rather to help the brain grow and adapt to changes, that sort of thing. So, does that stuff address what you're asking about regeneration? So, I mean, to just ask a question about regeneration is kind of hard to answer because there's so many different types of cells, and how they each go about doing it is tough. And I think laying it out in terms of tissue does illustrate a little bit about some of the major groups of cells and how they do their job. Any other questions? Okay. So in the presentations, it talks about the types of tissue. There are 4 of them. And I've done this presentation. It's available. You can see my brief explanation of the different types. I cover this. I have this particular picture in my presentation. I talk about it, but I want to spend a little bit of time explaining this concept in more depth, which is the ultimate sources. Oh, great, everything's going to be slow today. Okay, the ultimate sources of the tissue that we're talking about. Now, ultimately, everything comes from that initial fertilized egg cell. Okay. So a man and a woman get together, yada, 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 you have a fertilized egg cell. And if you don't know the yada, yada, yada part, go talk to your mom. But that single cell has all of the DNA that you've inherited from your mom and your dad, and the cell divides into 2, and they both have a complete copy of that DNA. And then those 2 divide each to give us 4 cells overall, each with the exact same DNA. And then there's another division, another division, another division, and the cells grow exponentially. So from 1 cell, you go to 2 to 4 to 8 to 16 to 32 to 64 to 128 to 256 to, where are we next? 512, 124, et cetera, et cetera. Now, if it seems very impressive that I can do all those numbers in my head, I really had to practice. And those are just the powers of 2, okay? Which is a fairly straightforward exponential progression. Somewhere around, I want to say it's the 500 cell range, somewhere in that number. The embryo, or what's going to become the embryo, starts to differentiate into 3 different types of tissue. There's the ectoderm, the mesoderm, and the endoderm. Now, derm, which you might say, hey, we were just using some words that have that in it, like epidermis and dermis. Derm really just means tissue. Okay, so for the skin, the epidermis, the outer layer of that tissue, and the dermis is the main layer of that tissue. But here we're just using it to talk about tissue in another sense. And ecto, meso, and endo are just describing the relationship of those 3 layers of tissue to each other. What does ecto mean?
Outside.
The body.
Yeah, outside. Okay. And what's meso mean?
Middle.
And endo?
Inside.
Inside. So that's all it is. Just you look at the wall of the developing zygote, what's going to become the embryo. The outermost tissue is the ectoderm, the innermost tissue is the endoderm, and between them is the mesoderm. Those cells, again, originally came from the single fertilized egg cell, but with each division, while each cell gets the exact same DNA, they don't use the DNA the exact same way from there on. That's what we call differentiation, where cells start to switch genes on and off to switch genes on and off, and so they start to behave differently. And eventually, through the differentiation process, these three different layers of tissue are going to become more specifically the mature tissues that we talked about in the adult. Ectoderm gives rise to the skin and the nervous system. And so it's going to give rise to skin cells, which make up most of the epidermis, for example, and neurons, which we're going to find in the nervous system, obviously. And then they mention specifically a pigment cell, and this is a cell that's in the skin. So all of these yellow colored cells here that are stacked up like bricks in a wall make up the epidermis. But at the basal layer of the epidermis, there are these specialized cells that make a pigment called melanin. And those come from the same source as the other skin cells and the nervous system cells. Mesoderm makes the various types of muscle, so cardiac muscle, skeletal muscle, and smooth muscle, which are all colored red here because they have a molecule in them that reacts with oxygen and gives them a reddish color. We have red blood cells to represent some connective tissues, and they are, of course, red because they have a molecule in them that reacts with oxygen and makes it red. Does anybody know the name of the molecule in red blood cells that turns red when it reacts with oxygen? If I was to ask you to name a protein in blood, what would be your first response?
Hemoglobin.
Right, hemoglobin. That's what it is. Hemoglobin carries oxygen, and it does that because it has an iron ion in it that reacts with oxygen. What do you get outside of the body if you put iron and oxygen together?
Rust.
Rust, iron oxide. What color is rust?
Red.
Red. What color is blood?
Right.
Red. For the exact same reason. There's iron oxide reactions taking place there. Now, the muscle's red for the same reason. They don't have hemoglobin in them, instead they have myoglobin. But it's a very similar protein with iron in it that reacts with oxygen. And so they have a reddish color to them also. And then for whatever reason, the nucleus of this tubule cell in the kidney is the same kind of red. I don't know if this was necessarily an artistic choice, but everything here is red, which I think is a little misleading. Now, it is true that you get muscle tissue and blood cells from that mesoderm layer, but you get other connective tissues from it also. And the tubule cell here represents an epithelium that's found in the kidney, obviously, but there are other epithelia that come from that too. There's a nice list of the different things that come out of that. So I don't know if that was necessarily an artistic choice, but I do want to point out in the skin and nervous system ones, there's this thematic use of the color yellow here at least. And to some extent that's typical. We use yellow to often represent nervous tissue. So in anatomical models, you'll see the nerves are in yellow, or collections of nerve cells out in the body called ganglia are represented in yellow too. And then it's just kind of every source that I can think of represents these cells from the skin in yellow too. I don't know if that's supposed to be because they come from the same tissue as the nervous tissue, but the color doesn't really mean as much in the skin. And then the endoderm gives rise to mostly the linings of internal spaces in the body. You have the abdominal cavity, the thoracic cavity, those sorts of things. They're lined with an epithelium. And then there are some endocrine glands that come out of that also. So, like the adrenal gland, and I suppose the pancreas and the, oh shoot, I was just doing this in AP2. Pancreas, adrenals, probably the thyroid gland or the parathyroid glands, those kinds of things. I actually don't know this, but I'm going to say the gonads, testes, and the ovaries. I'm not sure that's true. Oh, apparently not, because parts of the gonads are here from the mesoderm. But if you just think about a very, very simplistic body plan of the embryo, the skin and nervous systems growing out of the outside, and the linings of internal spaces growing out of the inside, and then everything between them coming out of the middle. So this is just giving a picture of where we go from the very simplest single fertilized egg cell to the very complicated adult plan, and where some of those tissues come from, and they're related to each other because of these embryonic origins that we see here. And to some degree, it's worth talking about that because in the connective tissues part of the chapter, it talks about the embryonic connective tissue, which is called mesenchyme. So it has that same prefix mes, like mesoderm. It comes from mesoderm, but we're a little bit further through development. And mesenchyme, for some reason I just like that word. It's a Greek word, but I like the whatever. This is embryonic connective tissue, and all other connective tissues are going to develop out of it. We can think of this as sort of the grandfather of connective tissues, and all of the rest grow out of it, or either directly or indirectly. Tendons and adipose tissue, which is fat, or cartilage, those things grow directly from mesenchyme. Bones are sort of indirectly from mesenchyme. Most bones develop from cartilage. The fetal skeleton starts as cartilage and then it calcifies later on. Or blood develops from red bone marrow, which is a type of connective tissue that comes directly from mesenchyme. So either mesenchyme becomes the connective tissues, or it becomes, or it gives rise to tissues that give rise to other connective tissues. So all connective tissues come from mesenchyme ultimately, some directly, some indirectly, but all of them can be traced back there, which is sort of a finer point from what I was just talking about before. Mesenchyme pretty much comes from the mesoderm, so all connective tissues come from the mesoderm. Not that that picture we just looked at made a good point of it, but that is true. I want to talk about sort of the scheme that we use for organizing tissues. And I want to talk about it for epithelial tissue and then for connective tissue, sort of as a framework for what we're doing with the lab part of this. So I'm going to skip over a number of things. I explained this stuff in the presentation. And then I do explain this picture too, but I want to highlight it a little bit because I think it's important to understand. When we're classifying epithelial tissues, we classify them based on two major criteria or characteristics. One is the layering, which is either a single layer of cells or multiple layers of cells, which we call simple or stratified epithelium. And then the other is cell shape. So there are flat cells, which are called squamous, cuboidal, meaning that in 3 dimensions they're kind of shaped like a cube. As we cut through a tissue like this, we see it in 2 dimensions, so they're kind of square in shape. And columnar, which means column shaped. And again, in 2 dimensions, they'd be tall, thin, like columns. Now, in recognizing these tissues, sometimes we can't really see the outline of the cell very well. And so the nucleus can be a clue. So squamous cells have flat nuclei. Cuboidal cells usually have round, centrally located nuclei. And columnar cells will have either oval or round nuclei, but they're not usually centrally located. They're usually offset somewhat. Like in this picture here, we see they're all sort of at the basal side of the cells, but they line up in a nice row. And when you see this in the microscope, it's really pretty obvious. You can see that row pretty easily. And speaking of, let me open something up. Okay, I want to try to pull up a picture. I don't know if it'll translate to the screen capture stuff that I'm doing online, but oh, that's right, everything's slow. Sorry, let me try that again. Okay. So this is one of the pictures that was captured yesterday of the tissue that we're looking at. And I'm pulling this up because I want to show you what I'm talking about, is a nice straight line. So this is the lining of the small intestine. And we can see this dark purple line that's kind of showing us where this epithelium is. It has a lighter purple to pink color sort of extending away from it. But the dark purple is a row of nuclei there. And oops. This is just the 40x objective. So here are those nuclei. They're all lined up in a nice row. The entire cell extends out here. When you're looking at columnar cells, I often describe this, you're going to see this apical cytoplasm. Okay? The cells have a basal and an apical side, the bottom and the top. And you'll know you're looking at columnar cells because there's going to be some clear cytoplasm beyond the nucleus. Now, the nuclei are in a nice little row, but they're all sort of so close together that you can't see them individually. But you can kind of imagine there's a nucleus there, there's a nucleus there. And so you can mentally draw in a line going up between those two nuclei and a line going up between those two nuclei. So we're talking about a tall, thin cell, okay? And a lot of the apical space is just clear cytoplasm. Okay, that's a columnar cell. Now, there's a special case for actually the simple and the stratified epithelium, and they've put the special case for the simple, but not the stratified in here. And that special case for simple, which means that it's a single layer of cells, but it doesn't fit exactly into the cell shape naming convention. This is called pseudostratified. Now, what does pseudo mean if we see that name?
False.
False, right. So this is a false stratified epithelium. And if it's not stratified, then it has to be simple. Now, the reason why it's a false stratified epithelium is that we don't ever see things quite as perfectly as this drawing suggests. So you can't actually make out the cells, but you can see the nuclei. And the nuclei aren't in a nice straight line like you see with the simple columnar. Instead, they're kind of at different positions. So it looks like multiple layers of cells. And so it can be confusing. People will think that it's stratified, but it's not. It's pseudostratified. All of the cells here go from the basal edge to the apical surface. Or really, what I should say, all of them start at the basal surface. The reason why the nuclei are in different positions is that there are cells that are growing in to replace old cells. And the ones that aren't full grown, their nuclei are going to be closer to the basal surface as opposed to the full grown ones that have higher nuclei. And in the presentation, I make this suggestion. Imagine you have a bunch of people lined up side by side. Some of them are adults, so they're full size. Some of them are adolescents, so they're a little shorter. Some of them are kids, so they're even shorter. But nobody's standing on anybody's shoulders, okay? So it's a single layer of people, single row of people. The heads will be at different heights because of how progressed each individual is through growing to their full height. Think of that as kind of why we have nuclei in all different positions. I have another picture I'll show you in a second that shows you the other special case. But imagine we had a fourth row here, and that fourth row was pseudostratified, and then the other one down here. It's maybe a better arrangement of this information. On the stratified side, okay, we have these three. The book kind of glosses over something with the way they've presented this, but stratified epithelia are named for whatever the top layer of cells is. And I've used this term a couple of times already, but we have an apical and a basal side to the epithelium. The basal side is the bottom of these pictures. And as they've drawn it in the book, we can tell which side that is because it always has this darker purple line. That's what we call the basement membrane. It's where the epithelium is attached to deeper tissue. The apical side is the top of the pictures, and there's nothing next to it. So epithelia are either covering or lining something. So they're going to be the last thing before we get to the outside of the structure, whether it's our whole body or an individual organ, or they're lining something. So they're the last thing before we get to the empty space inside, the lumen of something. So apical layers are always next to empty space on slides. And whatever cells we have in the top layer, the apical layer, that's the name for stratified stratified epithelium. So these are squamous cells, so this is a stratified squamous epithelium. These are cuboidal cells, so this is a stratified cuboidal. These are columnar cells, so this is stratified columnar. Now, it's maybe not terribly apparent from this picture, but the basal layer of cells in the stratified squamous example kind of looks like the ones down below. The nuclei are slightly different shapes, which is where it gets kind of ambiguous. But in a stratified squamous epithelium, you would actually expect to have other shaped cells in there. And the name just comes from the flat cells on the top. Stratified squamous epithelium is what we find in the epidermis. The basal layers of the epidermis are cuboidal to columnar shaped cells. And those are the cells that are dividing when I was talking about embryo. And this happens all the time. Okay, there's always some division going on there, and new cells get pushed up into higher levels. So if this cell right here divides, it'll divide sort of vertically, and one cell will become part of the next layer, and the other cell will stay on the basal layer. And then if it divides again, it'll push the new cell up, and the cell that just divided out of it will get pushed up even further. So the cells, as they regenerate naturally within the tissue, will just keep pushing older cells up and up. As it happens in the skin or in a stratified squamous epithelium anywhere, the cells end up getting flatter and flatter as they go higher up. And so the apical layer of cells always is going to be flat. They're originally cells that divided out of this basal layer, which is probably a different shaped cell. But by the time it gets to the top, they're flat. They're squamous cells. Stratified cuboidal epithelia almost always are, in fact, multiple layers of cuboidal cells, like this picture suggests. Stratified columnar epithelia is a very, very rare type of epithelium. What they've drawn here might be fairly representative of what it looks like. I'm used to thinking of the basal layer as actually being cuboidal cells, but sometimes cuboidal cells get a little tall, and it can get confusing. But we don't really care what shape it actually is. The upper layer is what's important. We're not going to bother with stratified columnar epithelium. There's not a good example in any of the slides that we have. So I'm just completely cutting it out of what you're responsible for as far as the lab material is concerned. It's not in the list of tissues that people sign up for. It helps because pseudostratified, if you're going to confuse it for anything, you're going to confuse it for stratified columnar. Okay. When I was showing you the simple columnar epithelium a second ago, and I talked about that apical cytoplasm, okay? In a pseudostratified epithelium, you're going to have that apical cytoplasm also. This particular cell right here looks like the nucleus is pretty close to the top of the cell, but really there's going to be a little gap between you before you get to the top. So a pseudostratified epithelium will obviously be columnar because it has that apical cytoplasm in it. But the nuclei are all over the place. So apical cytoplasm and nuclei in multiple layers could describe either of these types of epithelia. Okay. Could not describe any of these other ones, but could describe either stratified columnar or pseudostratified. So keep in mind, we're just ignoring stratified columnar. It's not, it's too rare of an epithelium for us to even deal with. So if you ever are looking at pseudostratified epithelium and you're thinking it might be something else, whatever else it could be isn't what we're studying. So it's got to be pseudostratified. Okay. Some books that I've used in the past will leave stratified cuboidal and stratified columnar out. We are not doing that. We're going to look at stratified cuboidal because we have a good slide for it. Sweat glands are stratified cuboidal epithelia, the ducts of sweat glands. So that's a good example of it, and we're already looking at the skin slide for stratified squamous in the epidermis and dense irregular connective tissue in the dermis. So we're already in that slide. It makes sense to consider that type of epithelium because it's there. But we're not looking at any slide that has stratified columnar epithelium in it because we don't have a reliable slide of that. Now, in the book, they have a table a little later on, which we see here, which I want to pull up. As far as the types of epithelium, it kind of says the same stuff that we just saw, except that instead of breaking it into simple and stratified columns, it's all one column with 8 different types. So we have the simple squamous, the simple cuboidal, simple columnar, and pseudostratified, all which were in that previous thing. And the stratified squamous, stratified cuboidal, and stratified columnar, which remember, we're not bothering with that last one there, stratified columnar. But then it also introduces transitional epithelium, which, let me zoom in on this a little bit better, is the special case for stratified epithelium. So pseudostratified is a special case of simple, and it really is columnar cells. They just look kind of funny. Transitional epithelium is a special case for stratified epithelia, but it's a special case because we can't classify it based on the cell shape. Okay? So the apical layer cells here are not flat or cuboidal or columnar. They're a special type of cell that's specifically for this epithelium. I've seen sources refer to those as transitional cells. Transitional epithelium is found in essentially one place, which is in the urinary tract. So it's mostly in the bladder and ureters. There's very little of it actually in the urethra, but right where the urethra is coming out of the bladder, there's going to be a little bit of it. We'll concentrate mostly on these 2 locations. The slide we have, that most people have in their boxes, is of the ureter. A few people might have the bladder, but it's not a great slide to work with. We will always look at transitional epithelium in this kind of configuration. It's relaxed state when the bladder or the ureter is empty. If it fills, the wall of the bladder or the bladder itself is going to expand, and then the wall is going to stretch out a little bit. And so these cells can be stretched, and these transitional cells will kind of be elongated and become flatter. We don't call them squamous cells in that configuration. They're still transitional cells, but they do change their shape to allow the bladder to stretch out here, or the ureters, or the beginning of the urethra. And the idea there is just you want as much space possible to store the urine until you're ready to get rid of it. But if you don't have urine to store, you want the bladder to take up as little space as possible. So the epithelium actually helps it expand a little bit. So that's a special case there. So if we look at this figure altogether, we see 8 types of epithelia. 4 simple and 4 stratified. And in each case, we have a squamous, a cuboidal, a columnar, and a special. Squamous, cuboidal, columnar, and special. We get 8 types of epithelia out of comparing layers and cell types, if we include the special versions. There is a 9th type of epithelium, which isn't represented in this picture, but the book does talk about, and that's glandular tissue. So right after the figure that lays out those 8 types of epithelia, it starts to talk about glandular tissue and endocrine versus exocrine glands and gland structure. This figure is not in the presentations because I don't care too much about this aspect of gland structure, but it does give you a little bit of a clue as to what's going on. And I also don't include the types of secretion. I don't think it's that important. I do have this picture as an example of, this is an oil gland that we see associated with a hair follicle called a sebaceous gland. So that's an example of glandular tissue. I wish they had a picture of an endocrine gland also to sort of compare. So in the presentation, I have a picture of the, I think it's a pituitary gland that I have, as an example of glandular tissue, endocrine gland tissue. So you can see both of those there. I also want to talk briefly about how we organize connective tissues. And again, there is a presentation that goes into this in a bit more detail, but I do want to mention this for one reason, because I want to, oh, shoot. This isn't a picture that I can open up another page, it's just a table, which I can zoom in on. Good. So when we're talking about connective tissues, there's the connective tissues proper, which are sort of the original connective tissues, and the reason why it's called connective tissue. They connect things together. Like dense, regular connective tissue is a tendon. Okay. Dense, irregular connective tissue is, among other things, the dermis, which is holding the epidermis to the rest of your body. Adipose tissue is found in the subcutaneous fat, stuff below the skin, and it's part of holding the skin onto the rest of your body. Areolar connective tissue is always found next to epithelia. Okay. It's in every single organ of your body, so it's everywhere, but it's always what's next to epithelia, because being a loose connective tissue, there's room in that tissue for blood vessels to pass through. And so any blood vessels that are feeding an epithelium, kind of like these conduits I was saying over here, that would be in the areolar connective tissue, which would be adjacent to the epithelium. Okay. So those are always next to it. Now I want to point out a little error in this table. Under dense connective tissue, they have listed regular elastic and irregular elastic. Not correct. There's dense regular connective tissue, dense irregular connective tissue, and then there's dense elastic connective tissue. Okay. And elastic connective tissue is just a variation on, I think, regular connective tissue, but it has a lot of elastic fibers in it. In the slide sign up, I was pointing out that one slide with the question mark next to it. A good example of dense elastic connective tissue is in the wall of the aorta. when your heart beats, it pumps out, 80 or so milliliters of blood into the aorta, which is not a huge volume of blood, but for the aorta, it makes the aorta stretch out. Okay? But then there's elastic in the wall of the aorta, so right after the heartbeat, that elastic tissue pulls back in and squeezes the blood, which helps to push it on to move through the circulatory system. So just, there should be 3 bullet points here. I need to try to get them to fix this. Dense regular connective tissue, dense irregular connective tissue, and dense elastic connective tissue. Now, regular and irregular is about how the collagen fibers are oriented in those tissues. Regular means that they're parallel, irregular means that they're not. And I explain that in the presentation. Now, in the book, they go to talking about supportive connective tissues. Not a lot of sources will use that terminology for this, but I kind of understand where they're getting at with this. In that only the connective tissues proper are actually connective tissues. They're the only things that actually hold things together. And so the book is trying to deal with that idea. And so they're saying, well, these are supportive connective tissues, which is, they basically make up the skeleton. I don't like that too much because it's trying to force the word connective onto those tissues, and they're not really connective. But they belong to this family because everything here comes from mesenchyme initially. And that's what ties them together. And then also they share characteristics, which is they have a lot of extracellular space that gives each type its properties. For instance, the dense connective tissues are pretty much all collagen, which makes them very fibrous and tough, like a tendon. The loose connective tissues have a lot of water that collagen and those sorts of things are dissolved in, and so they're kind of jello-like in consistency. Cartilage, its extracellular space, has a special type of molecule in it that gives it a very rubbery consistency. And bones have crystals of calcium phosphate, which makes it very hard. And then blood has plasma, which is mostly water. And so what's in the extracellular component really defines the properties of each one, but all connective tissues have that. And all connective tissues come from mesenchyme. So I don't really like classifying these as supportive connective tissues because it's the book trying to kind of force that into something that doesn't really apply. But they are supportive in the sense that they're part of the skeleton. Okay. And then fluid connective tissues is a great term for this because these are liquid tissues. Now, I don't really think lymph should be a connective tissue by itself because it's really just fluids from other sources and cells from other sources. It's not a tissue unto itself, as I see it. But other sources will lump that in with blood. But really the cells that are in lymph are blood cells. So it's not like they have their own kinds of cells. So that's how we kind of organize the connective tissues. Okay. Let me just, oh, that's right. One other thing I want to touch on real quick, going back into the types of tissue section. It's a little unfortunate that this is where it is because it makes more sense to talk about this after you've talked about tissues in general. And that is the tissue membranes. So in the levels of organization, we have the chemical level, the cellular level, and the tissue level, and then we go to the organ level and the organ system level, that sort of thing. Actually, there are sort of transitions between those. Okay? Between molecules and cells, we have macromolecules and we have organelles. And between cells and tissues, there really isn't any transition there. But if we take molecules and cells and put them together, we get tissues. Okay? And so like collagen's a molecule, fibroblasts a cell, those two together make up aspects of connective tissues. But then when we go to tissues to organs, there is an intermediate step there, which is called membranes. Membranes are groups of tissue that work together, and there are 4 major types of membrane. There's mucous membranes, which line the digestive, respiratory, reproductive, and urinary tracts. There are serous membranes that line the inside of our major cavities, the thoracic and abdominal cavities, and cover the outside of the organs there. So they're pointing at the lungs because there's a serous membrane around the lung that lines the inside of the thoracic cavity. There's also a serous membrane around the heart that lines that part of the thoracic cavity. And then around the abdominal digestive organs, which is the intestines, the stomach, pancreas, and the liver, and gallbladder, there's a serous membrane that covers them and lines the inside of that cavity too. And then there's the cutaneous membrane, which is just the skin. There's only one cutaneous membrane. And then another membrane, which is different from the rest, is called synovial. These first 3 that I mentioned, mucus, serous, and cutaneous, are all an epithelium and associated connective tissue. Synovial membranes are really just connective tissue. You may find sources, and I can't promise that the book isn't one of them, that'll say that there is an epithelium there, but there's not really an epithelium. It's just what is there is kind of hard to classify. It's really connective tissue cells, like fibroblasts, that sort of thing. But they seem to be kind of in a layer, and some sources will call it an epithelium, but it's not really an epithelium. So there are 3 epithelial membranes and 1 non-epithelial membrane. It's just connective tissue. And synovial membranes line joint cavities. And what's important is that they secrete a fluid that acts as a lubricant in the joints as you're moving. And what makes that fluid is the thing that some sources will call an epithelium, but really isn't. So just understand this one is different from the other 3. And I haven't posted the presentation part of this yet, but I do sort of set these 3 off from that one so you can see the differences there. And I will, when I get to doing the voiceover, be very emphatic about the difference between the non-epithelial versus epithelial connective tissues. The problem I have with this is I'm trying to find pictures of just those membranes, and serous is, synovial is easy to find, and mucus is easy to find. Serous membranes are hard to find good pictures of that I can use in my presentations that aren't copyrighted. And cutaneous will be easy to find if I just spend a little bit of time. I just got to find a picture of the skin. So what I've done, and it got kind of rushed here at the end, is I want to make sure I covered certain things in case I'm going to cancel class on Thursday. If I do cancel class on Thursday, the rest of the material for this chapter will be in presentations that you can watch. Hopefully I'll have it posted by Thursday so you can pretend to use last time to watch those. But if I don't, it'll be over the weekend. But I also have to get the skin one up because that's sort of the lecture material for next week. Remember to sign up for your connective tissue that you can take pictures of, and come prepared as you want to for Monday. The more you understand about that tissue and the better you can find it on the microscope, the quicker you can take pictures and the quicker you can get out on that day. So very unstructured lab. Just you come in and you take the pictures, and when you're done, you're ready to leave. So I'll see everybody hopefully on Thursday, if not on Monday. I will text people to let you know what the final call on Thursday is.
First of all, in the